Hello, life science students. Today we're going to be asking the question, what is the scientific method? You might not know it, but you should know. We've already done it. When have we done the scientific method? Well, do you remember the growing plants gizmo? When we did this gizmo, we followed the steps of the scientific method. You may not have known it, but you did. We also completed that student exploration guide, and each one of those questions you answered was part of the scientific method. So let's take a look at the steps. Here they are. Step 1, state the problem. Step 2, gather information. Step 3, form a hypothesis. Step 4, conduct an experiment. And finally, step 5, draw a conclusion. So let's take a closer look at the steps now. Step 1, state the problem. When you state the problem, you ask, what question do you need to answer? So in the Growing Plants Gizmo, we were asking the question, does the amount of water affect how tall the plants grow? That was the question we were asking. That was what we were trying to answer. So now we go on to step 2. Step 2 is to gather information. In this step, we research our topic and we also think about our real world experiences. In the Growing Plants Gizmo, we answered the question, what do you think plants need to grow and be healthy? Well, have you grown plants before? Most of you answered this question with things like water, sunlight, soil. So you have to have some background knowledge about plants and what they need to be healthy and to grow. And so you probably have some experience, and therefore you were able to gather some information about what plants need to grow and be healthy. Next, we move on to step 3, forming a hypothesis. We make an educated guess based on our prior experiences and research, and we try to guess what will happen. We're trying to understand, what do plants need to grow and be healthy, and how will the amount of water affect how tall the plant grows? So we form a hypothesis. Do you think the seeds need water to start growing? Well, if you don't give the plant water, then it will not grow. That is our hypothesis. It's a statement, okay? Our hypothesis is, if you don't give the plant water, then it will not grow. You're making a specific prediction. Remember, science is predictable. You can make a specific prediction, and that's what your hypothesis is. So once you've formed your hypothesis, then you move on to step 4, and you put your hypothesis to the test. In step 4, you conduct an experiment. So you create a step-by-step procedure to answer the question or the problem. It's important that in your experiment, you change only one factor of the experiment to test your hypothesis. During this experiment, you'll collect data using quantitative and qualitative observations. So if you remember, in the gizmo, you were asked to set up the pots like this. In pot A, you put a bean seed in, 2 lights on, 0 milliliters of water per day. Pot B, a bean seed, 2 lights on, 50 milliliters of water per day. And pot C, bean seed, 2 lights on, 100 milliliters of water per day. But why did you do this? Do you know why? Well, because you were performing a controlled experiment. So in this step, what you want to make sure you do is you test one thing at a time. And we call these our variables. Those are the things in our experiment. So the first type of variable you have is the manipulated or independent variable. The manipulated variable is the part of the experiment that is being tested, okay? In the gizmo, the thing you were testing was the amount of water. You were testing, how will the amount of water affect the plant? That is your manipulated variable. That's the part of the experiment being tested. The other kind of variable you have is the responding or dependent variable. This is the part of the experiment that's being measured. So what were you measuring in the gizmo? Well, you were measuring the height, the mass, and the appearance of the plant. You could have found that after you ran the trial, you should have been able to see the height and mass, and you should have also been able to see it in your data table in your gizmo. The third kind of variable that we have are controlled variables. These are the parts of the experiment that are purposefully kept the same. So think about the gizmo and think about what you kept the same. What did you purposefully not change? Well, in the gizmo, our controlled experiments were the type of seed, the number of lights, and we just used regular soil. If you remember, in all of the pots, whether it was pot A, pot B, or pot C, we kept one bean seed, we had 2 lights on, and we had regular soil, no fertilizer or compost. And between all 3 pots, we kept those the same. And so the reason we did that is to make sure that the only thing we were changing was the amount of water. Because if we say we added compost to pot A and we only added 0 milliliters of water, but for pot B and for pot C, we didn't add compost, but we did change the 50 milliliters and to 100 milliliters. Well, we wouldn't know if it was the compost that was causing the change or if it was the amount of water that was causing the change. So what's important is that for an experiment, you change one thing, just the manipulated variable, just like we did in this experiment. We only changed the amount of water that we added to each pot. And finally, your last thing that you need to do is draw a conclusion. So that means you analyze and interpret your data. And we ask the question, does your data support or reject your hypothesis? Well, this is what your data table should have looked similar to. In pot A, where you added 0 milliliters of water, your height of your plant was 0 centimeters and had a mass of 0 grams. The appearance should have been that there was no plant there. It did not grow. In pot B, where you added 50 milliliters of water, you should have had a plant around the height of 51 centimeters and a mass around 5 grams. The appearance was that this plant should have reached the end of the ruler. It should have been pretty tall and had lots of leaves. And finally, in pot B, you should have had a plant that got 100 milliliters of water, was 46.7 centimeters tall, and had a mass around 6.6 grams. And again, the appearance of this plant was that it almost reached the end of the ruler. So now we want to interpret this data. What does it mean? Well, we can see that in the pot that had 0 milliliters of water, the plant did not grow. All right. And so what does that mean? Well, what do plants need to grow and be healthy? That's the question we're trying to answer. Well, plants need water, right? When you don't give any plants water, when you don't give them any water, they won't grow. And that's what we see here. So we answer our question with our data and we prove our hypothesis to be correct. So we supported our hypothesis, which was if you don't give plants water, then they will not grow. We supported that hypothesis because what did we do? We didn't give the plants water. What happened? They didn't grow. And so our conclusion here can be that plants need water. And there you have it, guys. Those are the steps of the scientific method. Thank you for following along. Be sure that you submit this Edpuzzle when you're all done. Thank you. Bye-bye.